Okay, thank you very much, Andre. So I will talk a bit about progress in empirical consciousness research today. And as was mentioned earlier, we have this collaboration called the ConsciousBrainConcepts at the University of Oslo, which I started 2 years ago, together with people from philosophy and psychology. And the aim was to try to help analyze and hopefully help to improve some of the concepts in the field that have been disputed and somewhat caused some confusion. Of course, we can only do a little bit in that direction, but I'm interested in that and see if that might help. But this is not what I'm going to talk about today. I'm going to talk about the empirical part of consciousness research. And I think this is relevant because, as you know, the topic of consciousness is at the interface between philosophy, and it used to belong to entirely to philosophy for several centuries, but more recently neuroscience have come into the picture more and more importantly. And the consciousness research is highly relevant for a lot of different topics, including the clinical fields of neurology, neurosurgery, neuropharmacology, and sociology, but also for ethical questions, because the consciousness also touched upon the question of free will, which is synonymous with conscious free will, and therefore it's important for responsibility and thereby law. And also it is relevant for, of course, in recent development in artificial intelligence and robotics that will be discussed tomorrow in the meeting tomorrow evening. And last year we had a special discussion, a public discussion about the philosophy of mind and theories of consciousness. So I thought it was appropriate this year to focus more on the empirical side, on the neuroscience of consciousness. So, of course, like all other sciences, the science of consciousness also has its roots in philosophy. And what's interesting and nice in the recent decades is that it's been increasing crosstalk between the philosophy of mind and the neuroscientists. When I came into the field about 30 years ago, there was a much stronger division between these areas. So that's really nice. So all this philosophy, theoretical work, artificial intelligence, and so on is all good. But I'm only like to stress that, of course, this consciousness research is not only about this, but to a very large and increasing extent about empirical research, about natural science like neurobiology, biophysics, biochemistry, medical sciences, etc. And of course, the hallmarks of science is to generate testable hypotheses and test them in experiments and other empirical studies. And that's what we're going to talk about. But of course, the topic of consciousness is very special in the sense that, unlike most other parts of science, which are based on objective observations and experiments, that goes for, I mean, all the physics and chemistry, all studies of matter, our own body, and our own brain. The concept of consciousness it comes from subjective experiences, and therefore doesn't seem to fit into this objectively based scientific worldview. And there's a lot to be said about this. It's related to the hard problem and the unbridgeable gap, etc. But I only stress now that in spite of these difficulties get generated by this subjective aspect, it's very important to at least do our best to try indirectly to assess the conscious phenomena in an objective manner, and then thereby try to use empirical science to test theories of consciousness. as most of you probably know very well, this very important and famous example of empirical science, the general relativity theory of Einstein, was proposed by him in 1915 based on entirely theoretical considerations, and was then first regarded as a very bold but exotic and theory that had still not been confirmed by experiments. But he made some very interesting testable predictions, and one of the boldest ones were that he predicted that light from a star would actually be bent on this way from the star to the Earth, where it can be observed because of the gravitational field of the sun. For stars that have its light path close to the sun, and because the gravitational field will bend the space itself. And this radical idea could be tested during a solar eclipse in 1919, where two British expeditions, one to South Africa and another to South America, took photographs like this, where you can see faint images of stars close to the disk of the sun. And these measurements showed that the apparent location of the star had now been shifted because of the bending of the light by the sun, and in a manner that was almost exactly as big as predicted by Einstein's theory. And that was the real breakthrough of this theory. And that's why I mentioned it now, because these radical and counterintuitive predictions are extremely important when they can be tested like this. And I mentioned it because the situation in conscious research is that we still have not got any confirmation like this of any theory. This is really radical and counterintuitive. There are many really radical predictions, but so far all of them have been beyond what we can test by experiments. So that's a big challenge, to try to develop our experimental tools and methods in order to, in the future, test some of these predictions and decide which theory is closer to the truth, and thereby make real progress in the field. Of course, science has been confronted with the unexplained phenomena of many kinds through the centuries. Gravitation itself was a big mystery before Newton's time, but he was able to solve it mathematically, but also by making the very bold leap that he assumed that it could be influenced from one material object to another at a distance. So this action at a distance was previously regarded by most physicists as something spooky or unscientific, but Newton assumed this counterintuitive force, and thereby was able to make a lot of highly testable predictions that were verified. Darwin, on the other hand, didn't make very radical assumptions. He was able to a large extent solve the mystery of life, which was an enormous mystery, how this strange phenomena could come about, by almost common sense in the theory of evolution. And of course, it was helped along with all sorts of biology and cell biology, biochemistry, genetics, etc. eventually. But all of these sciences are more or less common sense-like. They didn't need to make any radical new assumption about the nature, and again, Einstein needed to make this bold assumption about the relativity of space and time in order to explain the very strange observation of the constant speed of light in all directions that was otherwise unexplainable. And he came up with this radical general theory of relativity. And even bolder steps had to be taken by Bohr and Heisenberg in order to explain the inner workings of the atom and in the micro world of the elementary particles. through quantum mechanics, then they have to give up even the basic idea of deterministic quantum causal interactions at the quantum level. And thereby they can explain a lot of phenomena with a theory that's worked beautifully, but it's very counterintuitive. So the question is whether consciousness is such a strange, even stranger phenomenon, perhaps, that may need even more radical revolutions in science in order for us to come up with a good explanation, or whether we can use biology's and Darwin's method to just do regular science and accumulate a lot of evidence until the problem is almost dissolved without any radical assumptions. So that remains to be seen and very interesting. Now we'll turn to some key concepts. We'll discuss what we mean by the basic term consciousness and distinguish between the state or level of consciousness versus content, and the responsiveness versus consciousness, reportable or access consciousness versus unreportable or phenomenal consciousness, which are somewhat distinct, and also intelligence versus consciousness. And of course, the latter is relevant for tomorrow's discussion about artificial intelligence and machines, if there could be consciousness ever arising in machines. So, but most scientists in the field would say that intelligence is something a radically different concept from consciousness, although they are related, but to what extent remains to be seen. So again, back to the basic concept of consciousness. Most people in the field use the very basic meaning of the term and define consciousness as equal to experience. What it is like to be a person or maybe an animal or whatever, the experience from inside. So to be conscious is to be able to experience something, anything at all. There's another definition of consciousness, and that is what is lost in dreamless sleep or general anesthesia or coma. And these are closely related, although not quite the same. And the experiences that are lost include all sensory experiences through perception, like seeing, hearing, tasting, smelling, feeling, etc. And our thoughts and memories and our emotions, like happiness, fear, pleasure, whatever, but also dreams and hallucinations and desires and wishes and will. And these latter experiences are, including emotions, actually are of course triggered or generated from within the brain, and does not correspond directly to anything outside in the outside world. And to include them is a very important step, because it means that also our dreams and hallucinations are really, per definition, part of our consciousness. And it shows that our brain, even when isolated from the outside world, can generate a whole world of experience that feels almost exactly like the real outside world experience that we have when we are awake. So that means that even maybe a brain in the vat can generate experiences, and it also implies that the world that we normally experience, like now when I'm awake and seeing the room around me, is actually a kind of virtual reality that is generated within the brain itself on the basis of sensory experience from the outside world, but is not identical to the outside world in any way, only indirectly related. But there is also another more clinical concept of consciousness that is different from the one we are talking about here. And that is more closely related to what we call responsiveness. But this responsiveness is not the same as consciousness. So in the clinics, it's important to determine whether a patient can feel anything, for instance, during surgery, during anesthesia, and or during and the degree of wakefulness when there's a brain injury. And you have this Glasgow Coma scale. It's a classical scale where it tests various responses of the eyes, the verbal responses, the motor responses, and then score these from 1 to 6 and come up with a sum that tells you whether the patient is deeply unconscious or fully awake, like normal, somewhere in between. But again, this clinical concept of consciousness is what we would call responsiveness. So there are, on this basis and based on other studies, it has been postulated that there are two kind of dimensions of consciousness. One is the degree of wakefulness or arousal, and along this axis, and another one is the content of the conscious experience. For instance, when you are awake, you can have more or less content, although you are always awake. And normally we sort of move along this diagonal where there's a correlation between the level and the content of consciousness. So when you are fully awake, you normally have a lot of content in our consciousness. When you are drowsy or in light sleep, you have less experiences, and in deep dreamless sleep we have less. In REM sleep and lucid or the time of dreaming we have more content, although we are have a lower level of arousal. And in general anesthesia and coma we are down here with very low level of arousal and also no content as far as you can tell. But in diseases and after injuries, there can be deviation from this, and then can you have a dissociation where you have some degree of arousal, open eyes, etc., but still very little content in the consciousness as far as you can tell. But we'll hear more about this from Olivia later today. So these levels of consciousness, they are closely related to what they call brain states of consciousness and unconsciousness, like sleep, anesthesia, and coma. So now, when starting to look at examples, we'll divide them between the ones that concern the global status of global levels of consciousness or arousal, and the more specific content. And we believe that, and have good evidence for that, the global level or states are mostly controlled from the brainstem and the basal forebrain, where there are these long fiber systems that activate the brain where you are awake, from the reticular activation system, and whereas the content is mostly contained and determined, we believe, by the so-called thalamocortical system, which are the more recently developed cortical and thalamic areas of the brain. So when picking these examples, I would have liked to pick examples that are both novel and with surprising and counterintuitive results, and that give new insights and also are important for different conditions and questions, and with potential practical usefulness as well. It's not always easy to find examples that fulfill all these, but I have a few that touch upon these aspects. First, starting on the global, with the global level and states. Then there's a central question: can the level or degree of consciousness be measured objectively? And that can be translated in one way, whether the states of consciousness can be measured objectively. And this is highly important practically, in particular for general anesthesia, for surgery, which are used in several millions of patients every year. And it's therefore extremely important to be able to measure whether they are really unconscious or not, because it happens from time to time that the anesthesia doesn't work and the patients can wake up. But before we go into this, we have to talk a bit about some leading theories or models of consciousness. These are the two I could say, most popular or most famous theories of consciousness is the global neural workspace theory, launched by Jean-Pierre Chancheux and Stanley Stahan in Paris, and it's the integrated information theory mainly developed by Giulio Tononi in Wisconsin. And these are two theories that are different in many ways. This theory starts in a more conventional manner from brain mechanisms, like other neuroscience theories, and then try to deduce and to find correlations of what are hallmarks of conditions that give rise to conscious content of experience. They find that when something reaches consciousness, there's a stimulus that is strong enough and important enough for the brain to cause an ignition of the whole brain network, and thereby engage even the nearly all parts of the cortical surface or the cortical system, including also the language areas, so the person can talk about it and tell what the experience were. The other theory, limitation of this theory, it could be said, is that it doesn't really explain why this ignition will cause conscious experience. How can this physical phenomena during this widespread strong activity actually cause experience rather than being unconscious? So this other theory take the other approach, a radical approach, where it starts actually with the consciousness itself and try to deduce from there what are its properties, and from there try to deduce what kind of system can support such properties. I will, it's a very complex and demanding theory, so I will not have time to go into that, but I just mention that from the theories, in principle, possible to calculate a measure called phi max. That sort of is a measure of the total consciousness content of the system. So I'll get back to that because of some of the theories, sort of the methods used experimentally have been inspired or derived from this theory. So this concerns, this value here concerns primarily the global consciousness, the whole total content of consciousness in the system, whereas these theorists have been mostly interested in the specific content, whether a certain stimulus comes to awareness or not. As I said, these two theories have been inspiring different methods. So in this tradition of the global neural workspace theory, they have focused much on these event-related potentials that can be measured by EEG, by brainwave measurements about the brain, and where they see kind of a late activity. Here's the stimulus here, and only after long latency can they see a wave that corresponds to what they believe is consciousness, and they can correlate the conscious experience. The interactive information theory is using a different method where they perturb the brain. But we'll hear more about this from Olivia later, so I'll not talk much about that. I just mentioned that this theory, this approach to quantifying consciousness uses a kind of perturbation of the system by injecting a brief magnetic pulse into one place in the brain and see how the activity spreads out in the brain. This was developed primarily by Marcello Massimini and Adnan Casal, based on Giulio Tononi's theories, and Olivia and Gosseris and Steve Lores and Silvia Casarotta have been all active in developing and testing this theory. So this measure actually measures the brain's complexity primarily. In other words, its capacity for complex processing, which is equated in the theory to the capacity for integrated information. That is the combination of integration of information and differentiation of information, how rich it is in content. And according to integration information T, this is actually the capacity of the system for consciousness. And I will not go into, I think I'll talk more about this. I only mention that when the person is, this is tested in humans extensively, and in an awake person that is conscious and aware, you give it brief magnetic stimulation here, then you see throughout the brain, these are recording from many parts of the brain, you see a very complex but deterministic response that lasts for many 100 milliseconds. Whereas during anesthesia or sleep, you see still a strong initial stimulus, but then the it's an initial response, but then the response dies out because it's very simplified, although it's big. So this corresponds to a high degree of complexity and consciousness. This corresponds to low degree of complexity and consciousness. And this number that can be calculated on the basis of how much the signal can be compressed is always above a certain threshold value of 0.3 or so, when the person is awake and aware, and then the person falls asleep or going into anesthesia, then the value drops down to below this critical value, and then the person is unconscious. And this is not taught, Aliva will talk about this, I think, but then this can be also used in the clinics to follow the development of consciousness in patients. I just mentioned that in our lab, Alessandro Arena, a postdoc in my lab from Italy, he has been able to do the same kind of measurements in rodents, in rats. And that has been tried by several laboratories throughout the world for more than a decade before, but without much success. But he has been able to do this with screw electrodes in the scalp, etc. And by giving anesthesia, he found very much the same as has been given in humans, where the response goes from highly complex to much simplified with different kinds of anesthesia, propofol and sevoflurane. And you see this index, PCI, perturbation complexity index, falls down in different kind of anesthesia. Now we'll turn to more tests of specific content of consciousness within each state, rather than the different states of consciousness like anesthesia and sleep and wakefulness. So one central question is, why do some stimuli evoke experiences that can be reported? We can talk about it and tell about it. In other words, why do some stimuli reach consciousness, whereas many other stimuli fail to reach consciousness? We know a lot of the activity on the brain is not causing direct conscious experiences, although the physical phenomena are the processes in there, like action potential, synaptic potential, etc., are very, very similar. So what is it about? What's so special about this activity that causes conscious reportable processes? And I should warn you that, of course, whether it's reportable or not is different from whether it's conscious or not. But for the moment, we'll just lump these together. A very famous and striking example comes from split brain patients, who are patients who have suffered from severe epilepsy that cannot be treated well with drugs. And neurologists and neurosurgeons found out that they can be helped by actually cutting, drastic operation to cut the connection between the two cortical hemispheres, but cutting the corpus callosum that connected to with millions of nerve fibers. Surprisingly, this the patients report that they don't feel any difference after operation than they did before. So they function in almost exactly the same manner, and it's very hard to tell the difference. So only careful testing by Roger Sperry, who got the Nobel Prize for this work, showed that these have some strange phenomena in their minds. For instance, if you flash a picture like this, with the one word here, ring, and another word here, key, then the person will pick up with his left arm only a key, but not a ring. But when you ask him what he saw, he will only say that he saw the ring. And the reason we think is that this picture is only projected into the left part of the brain, where the speech center is, so therefore he can tell you that he saw this ring, whereas the key word was projected only to the right part of the brain, and therefore that has now no connection with the speech center, so you cannot say it. Although we believe that this part of the brain is active and probably conscious and experiences to have seen this word key, and can actually, by directing the left arm, can pick up the key behind the board here. So I just showed you a brief video from Michael Gazzaniga, the collaborator of Roger Sperry. So this is Joe, who had surgery about 7 years ago, and what
left hemisphere and right hemisphere now
and as now
are working independent of each other, but you don't notice it.
So he says he doesn't notice any difference from before. But now we may take it fast forward, if we don't have time to watch the whole video, in a situation where he's shown this on the screen, two pictures. In a minute now, we'll show a picture of a saw and a hammer on the left and the right. He asked what he saw. He saw a hammer, he says.
Draw with your left hand.
But then he's asked to draw with his left hand. And instead of drawing a hammer, he now is drawing a saw, corresponding to this, what he saw on the left side. So this is quite a surprising result. And it illustrates that subjective reports cannot always be fully trusted. Although consciousness is about subjective experience, we cannot always fully trust the reports without interpreting them in the light of what we know about the brain. Another quite dramatic situation is the so-called VADA test. It's not quite so dramatic as the surgery, of course, but in this, this is done before neurosurgery in order to find out which side of the brain contains the language areas and most of the memory functions. And this is done before surgery, where one have to remove a small part of one hemisphere in order to relieve severe epilepsy or maybe a tumor surgery. And to test where the language area is, one injects into the artery in one side of the brain only a fast-acting anesthetic agent that will put this part of the brain to sleep while the other part of the brain is still awake. And you see here an angiographic photograph of the contrast dye that have injected into the arteries on one side. So this is the part that will be anesthetized, whereas the other part is then awake. And this is quite a striking situation also, because when this happens, and we have been part of this in the hospital, we have been watching this kind of procedure several times, and because we are studying this, we see that when, say, the right half of the brain is anesthetized, the patient, before the anesthesia, holds up his arms, and then the left arm will drop down because the left hand part of the body is paralyzed. But in spite of that, the patient reports that he feels just like normal, and when asked, he didn't notice that the left side was paralyzed. And that's because that left part of space is gone. It's called hemi-neglect. Not only are the sensory input for that part of the space not attended to, but even the concept of that part of space has disappeared when the right part of the brain is anesthetized. So it's very surprising, very hard to predict from theory. It illustrates the importance of doing this kind of tests. We want to use the test as to test theories of consciousness. I don't have time to explain how, but this has been in our plans, and we are building up for this for several years. So we'll get back to that later. In the meantime, we also tested some data from some patients like this, the brain wave recordings or EEG data. And the striking finding there is that although the drug is injected, anesthetic drug is injected only in one half of the brain, as I showed you, the EEG changes, they are widely distributed across both parts of the brain. It's much more symmetric than we would imagine, although the right half of the brain is still awake. It's still influenced by this the slow activity that's induced by the left hand anesthesia. So that in order to explain this, we have come up with a new suggested new principle that called the cross-state unreceptiveness, whereby I think that even if the other part of the brain is influenced by slow activity, the function of that part of the brain is not very much affected, so the patient still feels normal and is awake, etc. Anyway, we'll now turn to another study, elegant study of, that concerns conscious report or conscious experience of the content of consciousness. Now, I would like to mention another example about the content of conscious experience from a recent paper, very elegant paper by Peter Rolstemma in Amsterdam. He is studying monkeys and tried to find out why some stimuli, visual stimuli, can be experienced, whereas other stimuli that are of the same intensity are not experienced. Because if you give quite weak stimuli that are across the threshold for experience, sometimes they can be seen and sometimes not. And so the animal is sitting in the front of a screen here, and it's getting some tasty fruit juice as a reward to collaborate. So it's trained to fix its eyes in the middle of the screen, and then suddenly there appears some spots on the screen. So by the way, this technology is similar to what is used in humans sometimes. So the animals have implanted in the surface of the brain and into the cortex these thin needle electrodes that can pick up activity from the brain cells in the cortex, in different parts of the cortex. And this same technology is used also in humans to help people who are paralyzed and cannot move their limbs or even talk. And by training, they can use these signals that are picked up directly from the brain through these electrodes to simply by thinking to direct a mouse on a computer screen and thereby point to different letters, etc., and thereby spell out messages and communicate with the world. These are so-called Utah arrays, and these are very important tools to develop brain-computer interfaces whereby people who are disabled in this way can then still control both computers, robotic arms, etc. Unfortunately, that was not available when Stephen Hawking was paralyzed by ALS. He had a system that was different by using his eye direction, but not as advanced as this. He, as you know, died a few years ago. Anyway, back to the experiments. So that this study is based on signal detection theory, where a system, whether it's a machine or a human brain or a monkey brain, can detect a signal when it is strong enough to get above a certain threshold. So if the stimulus is stronger than the threshold value, then the person will say he saw the spot, and then it's called a hit. Or the animal will then signal that it saw the spot on the screen by directing its gaze, its eyes, towards another spot, or the same place where the spot appeared. And thereby one can know whether animals saw the spot or not. And but there is also some noise in all such systems, and this shows the probability distribution of the noise. And sometimes the noise will happen to be strong enough that it can also trigger a response. It's above threshold, and thereby and trigger a response. So the person will then, or the animal will then, feel that it saw something and signal that it saw something by moving its eyes. But this is then a false alarm because there was actually no real stimulus there, only noise. And on the other hand, the stimulus can also be, when it's below the threshold, is too weak to be detected, there will be a miss, so the animal does not report any observation. So again, the animal sits here with electrodes in his head. Then starts to fixate a point in the middle of the screen. Suddenly there appears a stimulus in one part of the screen, and after delay, this red dot in the middle becomes blue, and then it should direct its gaze, its eyes, to this spot where this used to be. So for you, if you now focus there, and then you suddenly see, so there was something down here. Then when this becomes blue, you should look down here, and then the direction of the eyes will be monitored, and then that will tell the experimenter that the animal saw the spot. And this can be plotted, whether it's detected or not, this spot can be plotted, the responses can be plotted here as a function of the stimulus intensity or contrast. So when it's a high contrast, it's easy, so then the hit rate is very high, of course, whereas if you are below the threshold, then it's very difficult, and then the hit rate goes down. At the same time, the researchers recorded, as I said, these electrodes, both from the primary visual cortex, where the signals from the eyes first arrive, and then higher order visual cortex, where the signals are analyzed, and also in the prefrontal cortex, where the evidence is summed up and the decision is made whether there was a stimulus or not. And this is signaled by a movement of the eyes. So what they found was that with different strengths of stimulation, you see from yellow to red here, the different degrees of response. And they see that in the primary and secondary visual areas, there is a fairly short-lasting response that lasts only a few hundred milliseconds, whereas in the prefrontal cortex, where the decision to move the eyes is apparently made, there's a very long-lasting activity. And this is thought to correspond to the so-called ignition phenomenon in the global neural workspace theory of Jean-Pierre Chancheux and Stanislas Dehaene. So whereas here you have feed-forward propagation of the signal from the back to the front of the brain, after ignition you have a lot of both feed-forward and feedback activation and almost a sort of resonant activity corresponding to working memory that is much more long-lasting and then enables the animal to act upon the stimulus and report that it saw something. And this, according to the theory, is when the animal sees something, it will generate its response that causes a report. So this observation seemed to confirm or go along with this global neural workspace hypothesis or model or theory from Chancheux and Dehaene. But they also found that when they looked closely at the responses, they saw that the hit responses in the earlier for difficult stimuli were smaller than some of the missed responses for the stronger stimuli when they look at the early part of the cortex. So this activity in the earlier part of the visual cortex did not predict the stimulus detection because there was a mismatch between the response size and the response and animals' behavioral response. In contrast, the activity in the prefrontal cortex, there the activity was always able to predict the conscious report. So here, when there was a hit, the animal reported that they saw the stimulus. The response there in the prefrontal cortex was always bigger than it was when there was a miss. So then there was. So this seems to indicate that the activity here is a better predictor of the conscious report than the activity in the early part in the back of the head. But this can be criticized in many ways, this study, although it's very elegant, because it's only this effect could be due to this is strongly related to the motor response itself rather than experience. So that's something we have to keep in mind. Anyway, they made different and try to make to explain why some of the stimuli were below threshold and some were above threshold for a report, or thereby probably conscious experience, by combining different parameters like the time to fixation of the pupil diameter and the change in pupil diameter and the neuronal activity and different brain waves, etc. And they found that they could fairly well predict the hits and the misses from this. But the bottom line of all this is that markers of the activity before the stimulus were able to predict the conscious report and the conscious content. So that means, although the animal was always awake, it might have been in different states of arousal within the wakeful state, and that was the reason why it sometimes saw the spot and sometimes didn't. Finally, I will talk a little bit about a radical new direction in consciousness research, where one tries to bring into the discussion the cellular mechanism of brain processes and how they could be relevant for consciousness. Although this consciousness is thought to be, as I said, a fairly global phenomenon, it's still, according to all both his theories and many other theories, it still depends, this global connectivity and interaction depends upon the local interactions even within the same cell. And that is what this theory about. And this is based on a very important observation that was made by Matthew Larkham, a good friend of mine and collaborator within the Human Brain Project, who back in the 1990s found that when he recorded from this, the largest, some of the largest cells in the brain, the 5 pyramidal cells in the rat sensory cortex, he could see that there was important signaling process within the same cell. So when he recorded from the soma and stimulate here far out in the dendrites, there's only almost no response in the soma, only a fairly small response in the dendrites. And when it stimulates only in the middle of the dendrite, there's also a fairly limited response both in the dendrites and the soma. When it stimulated both of these together, you got an almost explosive increase in the response in the soma. And then suddenly the cell sends out not only one action potential or nerve impulse, but the whole burst of action potentials that signals that something special has happened. And this is a very important discovery, it seems. And I was in Sakna's lab at that time because I was about to learn how to record from dendrites. When Matthew was there, he was leading, almost the world champion in this technique at that time, and still is in the forefront. But the importance of this discovery can be explained as follows. In these large pyramidal cells, they get different types of information in different parts of the cells. So nearly all or most of the sensory information from the outside world, which tells us about what's going on outside the brain, are fed in from the sensory organs in a feed-forward manner to the basal part of the brain, of these big cells. Whereas the distal part of the cell receives another type of information. It's a feedback information and the context information. But the crucial thing is that, according to the theory, this kind of information, all this a lot of detailed information, does not make any sense to the system or the cell until it's being interpreted and compared to this background information that comes from our memory, all that we have learned about the world throughout our life, all the relevant information from other parts of the visual field or other sensory modalities like hearing or touch or smell, and the higher or the thalamus, and also emotional states from the amygdala will influence our interpretation of this information. So only when this is compared and there's a good match between what you expect from all this background information and what you actually get of input, when there's a good match, then you see this strong burst output that tells the downstream system that here there have been recognition of something, and only then, according to the theory, can we experience what we have, what the sensory organs tells us. If there's no matching, then there's no experience. So when, for instance, we see, look at the cup, we get all this detailed information in a feed-forward manner to the bottom of the cells. We still don't understand it and don't experience it until it has been interpreted in light of all this feedback and background information. And this has been confirmed in experiments by Matthew Larkin's lab, by Takahashi and Larkin in the landmark paper in Science 2016, where they tested predictions from this theory by selectively manipulating several different ways, very clever ways, to manipulate the stimulation of the distal dendrites. And they found that always when they boosted or strengthened the stimulation in the distal dendrites, then, like here, then the threshold for the stimulus detection went down. And actually there were also some false alarms, as if the animals detected the stimulus without having experienced it. In this case, it was not seeing, it was touch to the whiskers of the rat. This was done in rats. Whereas when they inhibited this feedback information to the distal dendrites, then the threshold was elevated, so it took a stronger stimulus to detect the stimulus, for the animal to detect it. And so this rat are able to report that they see it by licking to the left or the right with their tongue. They are trained to do that. So this was a very strong confirmation of predictions of the theory, although it's not totally specific. But then came another study by Suzuki and Larkum last year that was published in a big paper in Cell, showing that also general anesthesia may affect this system. The comparison between the feed forward and feedback information. And what they found, to make a long story short, is that different kinds of anesthetics will then, so to speak, uncouple the distal dendrites from the proximal part of the cell, and thereby prevent the crosstalk and comparison between these two streams of information. So they used this very clever experimental approach, which I have not had time to go into. They use light stimulation of the distal dendrites and various type and extracellular field potential recording, etc. But you see, when there was in the awake animals was this strong strong burst of activity when there was a stimulus, and this was dependent upon input from the thalamus, that it causes this coupling to be strengthened, whereas in anesthetized animals, this coupling was almost shut down, so there was no response when the animal was stimulated. So this may explain also how anesthesia works, and it's a quite radical new model or explanation for anesthesia. And based on these observations, they have now been launched by Johan F, Suzuki, and Larkum, so-called dendritic information theory of consciousness, where they say that both these theories and several other theories, integrated information theory and global neural workspace theory, they depend upon intrinsic crosstalk within the cells. And when they cut this crosstalk or paralyze it, so to speak, with, for instance, anesthetics, and maybe the same things happen during deep sleep, then you lose consciousness because you lose this kind of processing. By the way, together with Johan F, we wrote another paper last year about dreaming, because I had this idea years ago, and it turned out that also Bill Phillips and Johan F had similar ideas. So we got together and wrote an article about how these mechanisms might provide a cellular mechanism of dreaming, although in this case it's not what they call apical amplification or strengthening by this input, but what they call apical drive, where the intrinsic input by itself can drive the output of the cells without any external inputs. Because when we are asleep, our eyes closed, there's very little input through the sensory pathway, at least is weakened. And at the same time, the neurochemical milieu in the brain is such that it will promote and enhance our input just from the inner sources or from our memory, etc. And then we can dream up an inner world based on the memory that are reconstructed in various ways. Whereas in anesthesia and maybe during sleep, you have hyper isolation where this thing doesn't work, and there is no almost no output, and there's thereby no experience. Anyway, I will have to stop there. I'm sorry, I'm over time already, and just remind you what Andrea just said, that we have this Forum for Consciousness Research here in Oslo, where we have had and hopefully will still get in the future very prominent speakers like David Chalmers, the famous philosopher of mind, Stanislas Dehaene that I mentioned from Paris, and Jean-Pierre Chancheux also, and Adrian Owen, Terence Deacon, Stephen Lores from Liège, and worked with Olivia and many others. And but so if you're interested, you can follow this, and you can hopefully we will have also electronic events in the future. This, by the way, is Giulio Tononi giving lectures about this theory here in 2014 here in Oslo. And I should also mention that we also had a big symposium in our international conference in the Human Brain Project in Barcelona 2 years ago, and only recently all the talks from that conference is out on YouTube. You can watch them there, both from Peter Alsema that I mentioned before, but also Olivia and others gave excellent talks there, so that you can watch. The quality is not perfect, but it was still a very interesting conference. So thank you very much for your attention, and I'll just have to leave it there, and I'll take questions afterwards. Thank you.